Good afternoon, everyone. Thank you for spending these few minutes with me as I tell you a bit about what we're doing with our Commonwealth of Massachusetts Spinal Cord Injury Trust research grant. My simple drawings on this flip chart are aimed at telling you how we are approaching finding out the molecules within the front end of axons, and I'll explain this, that might lead to transforming them from unable to regenerate after spinal cord injury to an ability to grow as if they were building the nervous system during development. So the brain, shown here from the front, the eyes, the cerebral cortex, has these red neurons that connect through their long arm-like axons down to neurons in the spinal cord to control the arms or the legs or bowel, bladder, or sexual function, and other neurons living in these orange balls who send out parts of themselves to send signals and bring them up into the brain. And as we all know, in spinal cord injury, it's not a problem of the neuron cell bodies themselves being damaged, but rather when the normally protective vertebral bones are crushed or injured, those axons down from the brain or up to the brain are crushed or severed or made to not function. So let me blow that up for you. And this cell body, the control center of a neuron, with its DNA in the nucleus and its dendrite input, has almost all of its volume in its arm, in its axon. And that can extend 1,000 to 100,000 times the size of this cell body. So this neuron here on my flip chart would actually send its axon a few football fields long. And if it were as large as my 6-foot-tall body, my arm would extend from Boston City Hall to Braintree or Marion or Providence, depending on what I was controlling, the face or the arms or the legs. And when neurons set up that circuitry during development or embryonic growth, these leading parts of the axon, the so-called growth cone, can grow through the young brain, through old brain, even through injured brain in many experiments. So those young growth cones can generate and regenerate. What allows such amazing growth? What are the RNAs and proteins and signals that we might like to use to make neurons regrow after injury? Well, in injury, those growth cones try to reform, but they ball up into a blocked so-called retraction bulb. They stop. But kudos to Zhigang He's lab, who we've already heard about, my close friend and colleague, who's been able to use certain treatments to get some of those axons to grow partially. But there are risks of those treatments. So if we could only look and study the growing or blocked or developing axons and find out what molecules are inside that allow generation and regeneration, that could lead to new treatments. And that's exactly what our grant is doing. So the big picture is we've come up with entirely new approaches to take out all the molecules from growing or blocked or trying to regrow growth cones in spinal cord injury. And this leads to a new ability to investigate the working end of the axon for spinal cord injury. And we're aiming to identify RNAs and proteins and signals that make axons grow as new treatment options in spinal cord injury. So again, I'd like to thank the Commonwealth of Massachusetts grant program. Thank you very much.